অল্প একটু মনে হয় স্যার বলেছেন।
Okay. Uh, Today we shall discuss on plant probiotics. Plant probiotics uh, are one of the important research agenda in our laboratory, and we have been working on this topic since 2010. And over the years, we have collected some very good uh, materials, and some of them have been also tested in the field. So let me show you one of my old lecture I delivered in 2018 in Hawaii, University of Hawaii. You can get some of the information here. Is it visible?
Yes, sir.
Okay. If it is visible, then I think it is relevant to your uh, review. Plant probiotic as tools for biorational management of phytopathogens. I delivered this talk as an outreach lecture uh, at University of Hawaii, Manoa. So some highlights of our discovery of plant probiotics as living biostimulants and biopesticides, and discovery of novel secondary metabolites and their inhibitory activities against phytopathogens. And later, I shall show you in another lecture our recent activities. Possibly you have already seen some of the probiotic bacteria showed promising effect in biocontrol of wheat blast. I already showed this thing to you before. Okay. This is a slide already I discussed, but I think it is better to discuss again. Plant probiotics or biologicals. Plant probiotics are microbes that live fully within plant without causing any disease, that provide multiple benefits, including nutrition and stress tolerance to plants. Here, stress tolerance means biotic and abiotic stress tolerance. So even in your review, you can add this definition. Uh, Endophytes are also called plant probiotics. And here I try to summarize the function, beneficial effects of plant probiotics. Here you can see uh, pathogen resistance is one of the important beneficial trait of plant probiotics. They can produce antimicrobial, not only antibacterial, antimicrobial compounds. Or antimicrobial compounds include small molecules like antibiotics or a larger molecules like uh, enzyme, lytic enzyme. And some of the molecules are specific for uh, metabolic pathway of the pathogen, not directly toxic, not directly inhibitory, but can impair the reproduction or uh, impair the metabolic pathway. So pathogen resistance uh, can directly be done by using antimicrobial substances. Other ways probiotic bacteria use to resist the pathogen and protect the plants are induced systemic resistance. That means probiotic can trigger expression of the resistant gene. and then plant immunity is boosted up. Uh, as a result, pathogen infection is restricted. But they also compete for the nutrient. For example, some probiotic bacteria can produce siderophores. Those compounds chelate the iron and deprive other microorganisms to get the iron. Uh, Even for making the biofilm physical barrier on the surface of the plant, they protect the plant. Biofilm, bacteria is like micrometer in size. You cannot see, but fully can cover the plant surface so that pathogen cannot get the space. So there are several ways I discussed now on plant pathogen resistance. Second is stress tolerance. Other stress tolerance, that means abiotic stress tolerance. For example, plants are sitting dark if there is high temperature, if there is drought, if there is nutrient scarcity, or even there is heavy metal toxicity. Plant cannot move away. Plants are sitting dark. In that case, uh, probiotic microorganism helps plant to tolerate uh, in the harsh environment. How? In most cases, they trigger the expression of the gene. In most cases, they uh, also produce different uh, antioxidants. In case of abiotic stress tolerance, abiotic stresses, plant cell produce higher amount of superoxides, and superoxides are toxic to the plant cell. So probiotic bacteria can enhance plants' production of antioxidants. Superoxides are like hydrogen peroxide, singlet oxygen. They are highly reactive. They are also called reactive oxygen species. They can damage the membrane, they can damage the DNA and affect the plant growth and development. In case of probiotic bacteria, they trigger plant to produce antioxidants, different enzymes that can scavenge or neutralize the superoxide or other metabolites that can react with the superoxide and convert them into safer molecule like water or like that. Stress tolerance is really an interesting area nowadays uh, for the probiotic application, because due to climate change, drought, high temperature, salinity, and many other problems are available, or we can see uh, that hamper plant productivity. In that case, even in case of salinity or in case of heavy metal toxicity, probiotic bacteria can, save the plant roots from those toxic substances. High salinity is really problematic for some plants, especially our cultivated plants. There are lots of mechanistic studies nowadays at the level of gene expression, at the level of nutrient uptake, at the level of in case of heavy metal, some of the probiotic bacteria hyper accumulator of heavy metal. They accumulate the heavy metal uptake and convert them into organic substances. and organic heavy metal are not toxic for the plant, because if it is soluble in ionic form, then it is problematic. Another point is remediation of pollutants. Already I mentioned, in case of heavy metal, you can use the uh, probiotic microorganisms to uh, hyperaccumulation and conversion of inorganic ionic form to the insoluble or insoluble organic substances, so plants save from the toxicity. Remediation, for example, many toxic substances for the plants, like uh, contamination of hydrocarbons or different pesticides, fungicides uh, in the oil, mining and other places, uh, polluted soil can be remediated by the microorganism, and some of them can easily degrade the toxic molecules to the non-toxic substances, and can remediate the soil. Other point is growth promotion. Growth promotion is one of the important target, and plant growth promotion are enhanced by the microorganisms residing in the rhizosphere as well as phyllosphere. They do this job by various ways. One is production of phytohormone. Over the years of uh, you know co-evolutionary interaction, uh, plant probiotic bacteria, through horizontal gene transfer or some other way, they can produce major phytohormones like uh, indole acetic acid, ethylene, gibberellic acid, or cytokinin and others. And they secrete and enhance the plant growth. They can fix the atmospheric nitrogen. Some of them even non-rhizobium, many uh, probiotic bacteria, they can fix atmospheric nitrogen and offer nitrogen nutrition to the plants, which is very important because we spend huge money for nitrogen nutrition to the plant by using urea and other fertilizer. So probiotic bacteria can do this job. Other job is that in the soil, most of the nutrient elements, among 16 or 17 essential elements, they are present in the soil at certain amount in agricultural soil, but most of them are uh, unavailable to the plants. They remain as organic form or inorganic insoluble form. Insoluble nutrients are not uptaken by the plants. Plant cannot uptake insoluble nutrient. Plant only can uptake nutrients in uh, available ionic form. Probiotic microorganisms can help plant to decompose the organic matter as well as insoluble inorganic substances to release nutrients in ionic form so that plant can uptake them. Growth promotion is indirectly be done when plants uh, are free from the you know insect and microorganismic pest. So when probiotic bacteria suppress the growth and proliferation of pathogenic microorganisms, as well as the insect, then it is a very good condition for plant growth promotion. Other thing is that plant probiotic bacteria can recruit and help the predator to attract the predator. So lots of ways plant probiotic bacteria can offer benefit to the host plant. Most of the plant probiotic bacteria uh, of our crop plants are not discovered. They are unknown. Because in the laboratory condition, we can only cultivate uh, maximum 5% of the probiotic bacteria or microorganisms. Most of them are uncultivated. Recent metagenomic study uh, is giving us a bigger picture. You may ask a question, if we do metagenomic study, how can we utilize the data? We know some of the genes, some of the enzymes that are useful for the plant. What we can do? We can utilize the genomic data for designing engineered culturable probiotic bacteria for the plant. Even gene can be transferred to the plant. So plant without probiotic bacteria, plant can do the job. For example, BT brinjal, BT brinjal contains a gene, cry1AC gene, uh, from the Bacillus thuringiensis. This is why it is protected from the Lepidoptera insect, brinjal, shoot and fruit borer. So this area is a very fascinating area, and we know very little about the crosstalks between probiotic bacteria and their host plant. And this is really an amazing area which could be uh, exploited for plant protection, plant growth promotion, stress tolerance, as well as remediation of the uh, polluted environment. And these are directly linked to the global climate change, mitigation of global climate change. This is directly linked to the eco-friendly agriculture and directly linked to the sustainable development goals. Many of the sustainable development goals uh, are linked to the application of probiotic microorganisms because they are safe. They are already present there. What you do, just modulate their population. And this area is an emerging area for research in agriculture. So uh, if you check the literature, please visit this paper, Amanda Rossier, Plant Molecular Biology, published in 2016. I'm sure you can find more details about the use of biologicals as biofertilizer and biocontrol agents for sustainable agriculture. The plant probiotics can be used as biofertilizer and biopesticide. I showed this image before. In our lab, since 2010, we have a visionary uh, project called Discovery of Plant Probiotic Bacteria uh, under an umbrella project, Mining Bangladesh Biogold. Bangladesh is not famous for, different minerals or gold or even fossil fuel. But Bangladesh is the largest delta in the world. It is rich in microorganisms, unique microorganisms. Probiotics are living microorganisms which when administered in adequate amounts confer a health benefits of the host. It is a general definition of probiotics. It can be human probiotics, it can be animal probiotics, it can be probiotics for fishes or plants. So the term plant probiotics has been used to describe plant-associated microorganisms which enhance the growth of the host plant when applied in adequate amounts, can be used as biofertilizer, can be used as a biopesticide, can be used as a biostimulant, can be used to enhance the abiotic stress tolerance. We have over the years we have developed many protocols, because to do and test the biological activity you need lots of protocol, and convenient protocol can help you to identify right probiotic bacteria as well as their biological activity in the laboratory, and then you can go to the greenhouse and finally to the practical field. So these are some of the, for example, a seedling assay. Here you can see without bacteria. In the petri dish, we germinated rice seeds. Rice seeds during germination, you can see like this. But here you can see when we added probiotic bacteria, dry matter production we have found uh, 3-4 fold increased without any nutrition. And that means some of the bacteria, probiotic bacteria, they are very powerful in enhancing the growth of plant, and obviously they can fix atmospheric nitrogen. Otherwise, dry matter production would not be so high. So it is an interesting subject to study in detail. They can produce the phytohormone, for example, indole acetic acid. Here you see some of the probiotic bacteria produce a high amount of indole acetic acid. They can inhibit the growth of the fungus. Here is the bacteria, here is the fungus. So due to the presence of bacteria secrete different antimicrobial substances, antifungal substances. This is why fungal growth is suppressed. Here you see what happened. The linear growth of the fungus is blocked because fungal cell became bulbous structure or lysed. We discovered some of the compounds produced by these bacteria. And we have developed lots of other techniques. Benefits of using plant probiotic bacteria provide nutrition of essential nutrient elements NPK to plants, NPK, zinc, and others, because they can uh, do the mineralization of uh, insoluble minerals present in the soil as well as organic matter that we apply or a crop residue to convert them into available nutrients in ionic form. Produce phytohormones and other signaling molecules of the plant. Control plant disease by antibiotics, competition, induce systemic resistance, production of lytic enzymes, uh, and uh, many more can be used as biofertilizer and biopesticide, which are alternative to the hazardous synthetic agrochemicals. And mines of novel antibiotics, enzymes, and other useful biomolecules. Over the years, if you see last 100 years, lots of antibiotics, enzymes, and biomolecules are discovered from the uh, plant-associated microorganisms or bacteria, fungi, and they are life-saving and billion-dollar business worldwide. This is why they are bioprospecting. That means you can develop an industry. In our case, we applied in many cases the application of probiotic bacteria. Here you can see uh, can protect the cucumber plants completely from the infection. Artificially, we applied the juice pool of Phytophthora, but in case of no probiotic bacteria completely died. In strawberry, we had experiment with a USDA funding project, and we found that strawberry production can be increased 43%, and the antioxidant in the strawberry also significantly increased. And a new antibiotic we discovered. Over the years, I discovered more than 50 antibiotics from the, probiotic bacteria. So this is, I think, some example. Discovery of plant probiotic as fertilizer. Major sources could be seeds, stem, root, and rhizosphere. And in our laboratory, we collected 48 locally improved varieties from the Unandara uh, to get the probiotic bacteria, endophytic bacteria in the seeds. And we, isolate lots of probiotic bacteria, and some of them we tested in the laboratory. And this figure already I described. Here you see the control and other with other bacteria. Some of them even not growth promoting. Some of them even suppress the growth, produce the phytotoxic compound. Those are not endophytic, because endophytic one always growth promoting. Evaluation of probiotic bacteria on growth and yield of rice by root colonization and spray application during flowering in pot experiment. In the pot experiment, we tested probiotic bacteria with probiotic bacteria, without probiotic bacteria, with probiotic bacteria and half dose of fertilizer, and full dose of fertilizer, and without probiotic bacteria, half dose of fertilizer, and full dose of fertilizer. What we found that in presence of probiotic bacteria, you see a full dose of fertilizer and half dose of fertilizer with probiotic bacteria, almost no difference. And this data you can see. With probiotic bacteria, what we found, you see the different probiotic bacteria, full dose fertilizer, the grain yield per pot, half dose and zero dose. Naturally, zero dose is small. But here you see in control, no probiotic bacteria, full dose of fertilizer, we received 12.98 gram rice seeds in a pot. But when we use half dose of fertilizer, recommended fertilizer, NPK fertilizer, and with bacteria, here you can see many of them are higher than the uh, half dose, higher than the full dose of fertilizer, even much more higher than the 1.6 times or 5 times higher than the full dose of fertilizer. That means, but if you compare with the full dose of fertilizer and probiotic bacteria, effect is not so robust, because in case of full dose of fertilizer, plant gets nutrition lavishly, and probiotic bacteria also gets those nutrition from the soil. This is why they are not functional. But in nutrient strive condition, half dose of fertilizer, they do tremendous job. That means we can save fertilizer 50%. Here you can see the half dose of fertilizer, full dose of fertilizer. And with this is control, and all others are, a different bacteria. Here you see half dose of fertilizer with bacteria BTS2 gave incredibly higher yield. And this means 50% of NPK fertilizers can be saved. And when we did the experiment in the greenhouse, we thought it is better to test in the field. And we also checked when we apply the probiotic bacteria whether they are colonized in the uh, rhizosphere rightly or not. Here you can see this is a scanning electron micrograph. Acinetobacter, when we applied in, you see all dots are bacteria, and they colonize and dominate on the surface of rice root. And this is why they do the robust impact. And we tested in our field, BSMREO field. Here you see probiotic control, full dose of fertilizer, and here half dose of fertilizer. Uh, 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 this is the control full dose, and here with probiotic bacteria. Morphologically, you cannot differentiate, but here even better. And by using Serratia and half dose, Micrococcus half dose, and recommended full dose. And if we see the quantitative data, here you can see uh, the full dose, BTS2 half dose, and BN8 half dose amazingly higher. And that means uh, BN8 Serratia marcescens really powerful. And we also did the whole genome sequence of Serratia, and we discovered that it can fix nitrogen and it has lots of plant growth promoting trait. This is why it is doing the job. And we also estimated if we can save the 50% NPK in and the rice cultivation, then billion taka can be saved, and it is it has high potential for commercialization. And we also checked it in the uh, wheat also, and in the wheat similar results we found. Here you can see the dry matter production. So uh, it this bacteria, when we applied in uh, strawberry, we found 43% higher yield. And we published our paper in scientific reports, Nature Groups Journal. Plant probiotic bacteria, Bacillus and Parabug holderia improved growth, yield, and content of antioxidants in strawberry fruits. Content of antioxidant incredibly increased, you see. And this is the control with probiotic bacteria. And antioxidants, especially, phenolic compounds, they are very rich present in the strawberry fruit. And strawberry fruit we take directly fresh fruit uncooked, so we get nutrition. Even vitamin C we found higher. So here you can see lots of other data, different kind of antioxidant like anthocyanin or carotenoid, flavonoids content, total phenolic contents, total antioxidant activity significantly increased. And this is the first report that fruit antioxidant content can be increased by the application of probiotic bacteria. But we don't know exact the pathway. Obviously, probiotic bacteria uh, enhance the gene expression for the biosynthesis of this compound, because these compounds has the properties of plant defense also. Uh, so this is the summary for today's lecture. We have a good bank of beneficial probiotic bacteria. Some of them are whole genome sequenced, and rest of them we will sequence soon. And mechanism of the plant growth, promotion, and disease suppression by these bacteria are partially elucidated in our lab. Uh, and obviously, if we can apply in the field, they can save huge money, as well as the fertilizer, synthetic fertilizer uh, production uh, requires raw materials like methane gas, for example, for urea. Huge methane gas is required, and greenhouse gas emission through the process is released. So we can mitigate the global warming by the application of probiotic bacteria. Another advantage is that probiotic bacteria are uh, renewable resource. If you have one plate or one uh, simple vial appended to you, few bacteria, as much as you can multiply, and it is never ending. Incredibly powerful natural resources for renewable use. But in case of synthetic fertilizer and other synthetic materials, you cannot use like that way. So this is why they are highly promising. But in the practical field, the application of probiotic bacteria is still limited, around $4 billion business worldwide. In our country, there is almost no industry. This is because our knowledge is still limited. We need to know in detail so that we can overcome the inconsistent performance of these materials in the field condition because they are sensitive, they are live. You have to maintain the shelf life of them. Farmers need to maintain in their environment. But we are focusing on the bacillus. Bacillus can produce the spore, which can survive even in the 80 degrees Celsius temperature and in good condition, good environment. When you apply, they germinate and produce and divide very fastly. Another point is the ecochemical relationship of the probiotic bacteria and the host plant. So if you isolate a bacteria from the rice, there is no guarantee that it will work on wheat because colonization on wheat depends on evolutionary relationship between the organisms. Some of the probiotic bacteria, they are a bit versatile and wide host range, but some of them are limited. But in case of industry, industrial people, or even farmers, they don't know it. This is why, like urea fertilizer or any pesticide, they want to use in the all crop. So we need to know much more about their host specificity, host range, as well as their sensitivity to the environment, soil pH, or soil other physical chemical properties, even temperature and environmental condition. And if you know all these things better, then you can design a product which could be useful even in a specific condition very reliably. Thank you so much for today's talk. We shall discuss more on the next lecture. So you can ask some question.
Recently, sir, I read a paper, and Spingomonas melonis is also helpful for or beneficial for the wheat. And I have another question. Sometimes endophytic bacteria are also in latent condition in the seed, but they are not acting. Um, Maybe they are in latent condition.
Let us
We,
start our
we can study. We can
next
also study in this area.
discuss on a new topic. I hope you are working on your. Yeah, yeah, please continue your question.
Yeah,
Shafi.
yes, sir. Yeah, yes, sir. Sometimes endophytic bacteria are also in seed and in latent condition.
Hmm.
Uh, and latent condition, but we don't know how they are working and what the condition is necessary for that endophytic organisms.
Yes, it is true. For example, you have started to work on seed endophytes in some genotypes of wheat uh, and to get their relationship with the blast resistance. We have no idea whether they are related or not, but we already discovered some bacteria from the wheat seed that in vitro suppress the wheat blast, in vivo, even in the field conditions, suppress the wheat blast. Our assumption is that seed contains diverse bacteria and fungi, as we have contrasting resistant genotypes, completely susceptible and completely, fully immune, there is a high chance of getting the probiotic bacteria that could be utilized as a commercial product. Most important is you told me the latent condition. Yes, they are present in a diverse, taxonomic, diverse lineage. But what is important, if you compare the metagenomes of uh, microorganisms, for example, bacteria present in the contrasting genotypes in their seed, you can get a picture. Obviously, you will get the diversity. And as most of the bacteria hugely been studied, comparing with their genus and species name, we can have some assumption that which are the major player may be potential for blast resistance or um, playing role in um, stress resistance of the plant in the practical field. Uh, and you will also check them in the living plant, that is leaves, as well as the spikes. And if we find them, those present in the seed, they are also some of the group present in the leaves or even in the spike, then we can target those to isolate in the laboratory. As we have the pathogen, we can check them in the laboratory quickly whether they have any beneficial effect in suppressing wheat blast. Wheat blast is one target, and if we get the bacteria, we can see also whether they have any effect on plant growth promotion or other beneficial effects of the plant. So, first, I would like to know the full landscape of the bacteria and fungi present in the seed, whether they are moving to the leaf, whether they are moving to the spike. Some of them you cannot find, maybe present in the seed, but cannot find in the spike because all are not, uh, how to say, all are not philosphere colonizing ability. Some of them colonize in the rhizosphere. This is why please also include the root. Then you can find some of them uh, present in the seed. They are colonizing in the root and helping plant to grow well and play a vital role in disease resistance also through induced systemic resistance and others. So please include root also. And we can find a global picture. After global picture, piece by piece, by isolating some of the members, like Spingomonas melonis, we may find an important member which have high potential to suppress the wheat blast and really play a vital role. And if we can manipulate its number through the seed priming as well as spraying in the field, maybe we can find a new solution of wheat blast. So your job, I think the journey is well defined, step by step where you proceed, and lots of, I think, new information you embrace, you can publish, as well as you can go for the product. Any question? What do you think?
And no, sir. Yeah, it's a nice research. I'm really interested, and hopefully I will enjoy.
So one thing I want to add now, uh, we need to do two things together. One is you are going to extract the DNA and do the metagenomic study. Parallelly, using the same sample, you may go for isolation of the bacteria. As you have only four genotypes, it would not be a big job. Using, crushing the seed after surface sterilization, crushing the seed and making a dilution series. You just spread on the, nutrient agar plate, and next morning you can find lots of, colony. Pick up the colony. Uh, Ruxana, do you know Ruxana?
Yes, sir.
Yes. So Roxana would help you to isolate the bacteria from each step. So pick up the bacteria, and Roxana would help you tremendously, as well as you can also do the job. Would not be big job to collect the bacteria in the Eppendorf and put in 20% glycerol and preserve in the refrigerator. So when we uh, uh, complete the metagenomic work, then we can, search that big collection, and then we shall concentrate on the bacteria, whether metagenomic study gave us whether we can get the same bacteria in our culture collection, and we can do the biological study. So in parallel, it is a hard job, but in parallel, if you can isolate or engage someone to isolate, Ruxana part time she can do even evening. I shall discuss with her. So please collaborate with her. If she has not a huge job in the NIMS project, she is skillful, and if she shows you, maybe every day you need to spend 1.5 hour job to do the job, not every day when you actually extract the seed, and maybe weekly 2 or 3 hours you can collect the bacteria, and that bacteria would be a treasure box, and we have minus 80 degree, 86-degree freezer. So what you can do, you can simply preserve them in glycerol, 20% glycerol, and after the metagenomic study, when there will be no wheat in the field, because you have now not only seed, you have the leaves, you have the spikes, so you can isolate bacteria from or root, and those would be the big, gold mine in future. Is it possible?
Yes, sir. It's possible.
If possible, then Nuruddin Mahmood, I shall ask him because he has no specific project. He will spend some of the time. Ruxana will also do some of the time. So please collaborate with these two persons, those two persons, so that they can help you to isolate the bacteria in every step of the DNA, every case of DNA. For example, from the seed, you collect parallelly the bacteria. From the leaves, you collect parallelly the bacteria. From the root, you collect parallelly the bacteria as well as genomic DNA, and from the spike. I think that would be the greatest strategy. And just store. No need to go for as much as possible single, isolate. First, you pick the single colony, which is unique, and you see no color combination. And then on the plate, I have shown you stick culture, and then again pick up the colony, and then preserve some colonies in the small Eppendorf tube at 20% glycerol. They will remain, uh, you know, live. And after this winter, when you complete the extraction of all DNA, at that time, you can slowly start studying their biological activity in the laboratory. right? Even I can engage new student like Joy Devnath to help you to do the laboratory job or other student.
Yes, sir.
So if you do parallelly, then it will be more authentic because same sample, you isolated the bacteria and you also identified by using bioinformatic tools and sequencing. That would be fantastic. Otherwise, you cannot make the story. Any question?
No, sir.
And thank you so much. So what you need to do, please sit with Mahmoud first and talk about my idea, and then sit with uh, Ruxana, and you may discuss with me because Ruxana has a job in another project, BUS USDA. But even though I shall offer her small funds for this part-time work if you can engage her. Clear?
Yes, sir. Clear.
Okay. We can offer her hourly and keep it, confidential because every day she will not have enough job for the NIMES project. Thank you so much. If you have no question, then have a good day.